All right, it is noon, y'all. Welcome. Good afternoon. So excited for today's talk. And I have the pleasure and honor of introducing Dr. Alexandra, Sullivan, who's a postdoctoral scholar at UCSF conducting interdisciplinary research on the biological embedding of stress and how positive parenting can protect children from the physical and psychiatric problems associated with early life adversity exposure. Dr. Sullivan earned her PhD in clinical and developmental psychology from the University of Vermont and completed her clinical internship at the Medical University of South Carolina. With 25 publications, 11 of which were first authored, and a track record of NIH funding through an NRSA, Dr. Sullivan is a productive junior scholar publishing in high-impact outlets such as psychological science and behavior research and therapy. Her novel work combines psychosocial parent-child interventions and child biomarkers reflective of transdiagnostic health outcomes. Her program of research has received recognition from organizations including the APA Division 53 Student Research Award and the American Psychosomatic Society Young Investigator Award. Today, she conducts research under the guidance of Dr. Nicole Bush through the Intergenerational Developmental Health Program and the Environmental Influences on Child Health Outcomes, ECHO, Pathways Study. It's so great to have you here, Allie, and please join me in a warm welcome.
Hi, everybody. How's this sound? Too loud or good? Okay, awesome. Thanks so much for coming today. I'm really excited to share with you some of the research I've been working on that I've called Unleashing the Power of Connection, Effects of Psychosocial Interventions on Child Biomarkers in Caregiver-Child Dyads. So in this talk, I'm going to describe a line of research that I've been working on investigating how sensitive parenting may support child health in kids exposed to adversity. I'm going to go through two distinct intervention studies testing dyadic intervention effects on accelerated aging. One examines how improvement in parenting interacts with adversity in kids who went through internet-based parent-child interaction therapy, and another looks at intervention effects of child-parent psychotherapy. But first, let me just give you a little background so we can all be on the same page when I go through these studies. Early life adversity gets under the skin, and it relates to future health problems. In response to stress, kids will mount a coping response that allows them to cope with environmental stressors. So in kids who are exposed to trauma, we might notice that they are more cued into danger signals, and are better able to potentially identify things like anger cues. This is really helpful. It helps them stay safe and protected in stressful environments. However, this upregulation can come at the cost of durable physiological changes and retuning their actual biology that then might relate to future health problems. This is particularly problematic for kids of color who are systematically exposed to elevated levels of adversity and consequently end up experiencing higher levels of health problems. Adversity exposure isn't deterministic, however, and there are things that we can do to help these kids. Prevention would be ideal, obviously. However, in cases where that's not feasible, we turn to intervention. Children's relationship with their caregivers is among the most potent of stress buffering resources available to them. So interventions that actually support sensitive and responsive caregiving may protect kids from the harms that adversity can cause. In this talk, I'm going to talk a lot about dyadic interventions. So I want to take a moment to define that for everybody. So a dyadic intervention is a programmatic effort to target the parent-child relationship and promote early relational health, that construct that can be really stress buffering for kids. It's rooted in attachment and social learning theories, and these programs leverage sensitive, nurturing, and adaptive interactions between child caregiver dyads to improve child outcomes. Typically, dyadic interventions are used to improve child behavior problems or trauma symptoms. And here are a few examples of different dyadic interventions that we see often in the community, like child-parent psychotherapy, PCIT, Triple P, helping the non-compliant child. So one reason we think that dyadic interventions improve children's mental health is potentially through paths of physical health. Supporting effective parenting practices that bolster attachment actually helps kids regulate better. And this improvement in regulation may alter the biological mechanisms that then, in the future, account for child physical and mental health problems. For example, in a recent review that I led with my UCSF colleagues here, Drs. Amanda Naronia-Zhu, Rubinov, and Bush, we found that dyadic psychosocial interventions improve children's cardiovascular functioning, advance brain maturation, and there was some evidence that is beginning to show that it actually decreases levels of inflammation and changes intracellular processes. Collectively, it seemed like interventions may be starting to have a whole body effect of improving child health. That's physical in addition to relational and psychological. Another important thing to think about here is that sensitive and responsive caregiving is helpful across development, but there are times during which it is particularly helpful for kids. Exposure to adversity early in development is particularly related to biological consequences. So it may be the case that exposure to these interventions early development is particularly stress buffering, including in buffering those biological consequences of adversity exposure. There are a million different ways to measure biological consequences of adversity, and today I really want to drill down into one. I'm going to talk more about epigenetics and the epigenome, and I want to take a moment to take a step back and understand what I'm talking about here. So epigenetic markers constitute one method in which scientists might be able to ascertain the biological embedding of stress. There are many different environmental influences that can shape someone's epigenome, like nutrition, exposure to chemicals, but perhaps the least studied of them is social experiences, like psychological stress. So epigenetic alterations are these long-lasting and durable processes that affect how cells process, how cellular processes activate, regulate, and express genetics or someone's underlying DNA sequence. Well, there are several different epigenetic processes. I'm going to focus on one today, which is called DNA methylation. This is one epigenetic process that is involved in gene regulation. So exposure to different social experiences, so exposure to stress, or maybe even exposure to positive parenting practices, result in tags on different locations of someone's epigenome. Collectively, these tags function as a light dimmer switch for gene regulation. So they either turn genes on, they turn them off, or somewhere in between. These differences in genetic expression then translate to differences in cellular processes, which then translates to differences in behaviors and in health. So that is to say, exposure to different social experiences may durably alter someone's DNA methylation and thus genetic expression, thereby associating with different health consequences down the road. So tools that measure epigenetic modifications, like DNA methylation levels, may be sensitive to different forms of trauma exposure and might also be a tool that we could use to tell what interventions are doing to kids' bodies. This brings me to a biomarker called the epigenetic clock. The epigenetic clock is a summary score of DNA methylation levels at specific sites on someone's epigenome that is reliably associated with someone's chronological age. So DNA methylation levels are used by taking biospecimen from kids, so a cheek swab, blood, saliva, and these DNA methylation levels are assayed from those specimens. And then we use a computer algorithm to calculate someone's epigenetic age from those DNA methylation levels. While epigenetic age and chronological age tend to be pretty correlated, they aren't exactly the same. And that variability and the strength of the correlation actually seems to relate to variability in health. So in this figure behind me, that dotted line on the diagonal represents the situation in which someone's chronological age is the same as their biological or epigenetic age. When someone's epigenetic age is older than their biological age, so a 6-year-old, and we take a swab of their cheek and we discern that their epigenetic or biological age is actually 7, this is the situation that we would call epigenetic age acceleration. And this is a situation where we notice that acceleration is associated with health problems. Growing literature seems to suggest that a faster ticking clock leads to poor physical health in kids. We've got evidence that accelerated epigenetic age is associated with childhood internalizing disorder diagnosis, PTSD diagnosis, reduced neonatal brain growth, higher BMI throughout adolescence, and more allergies and asthma. So it seems that accelerated epigenetic aging is a marker of potentially poor health. Along those same lines of thinking, adversity exposure actually seems to accelerate epigenetic aging. More and more research is showing this association. In a recent review of 69 studies, almost all of the studies documented positive associations between adversity exposure and accelerated biological aging in pediatric samples. Adversity was measured in a bunch of different ways, but some examples are maltreatment exposure, racial discrimination, adverse childhood experiences or ACEs. So what we're seeing is a trend that more adversity exposure seems to accelerate biological aging, at least that which is measured by the epigenetic clock. So if adversity exposure seems to accelerate aging, and accelerated aging seems to forecast poor health outcomes, the epigenetic clock might be a really useful intermediary phenotype for us to better understand differences in stress exposure and maybe differences in intervention exposure. It might be a really useful biomarker for us to quantify the biological effects of our interventions, especially our psychological ones. The remainder of the talk, I want to explore this question. So do dyadic interventions protect kids from accelerated aging associated with stress exposure? And I'm going to go into findings from 2 separate samples and 2 separate studies that seem to be suggesting this pattern of effects. So first, I want to discuss some findings from a study I conducted using a well-known intervention, parent-child interaction therapy. And this iteration of parent-child interaction therapy is particularly cool because it was an internet-based version, very relevant in today's era of increased remote access to treatment. I know a bunch of you are clinicians and have probably heard of PCIT, so I'm just going to briefly touch on what it is so we can all make sure we're on the same page. So parent-child interaction therapy is a manualized parenting intervention initially intended to reduce child behavior problems. So people usually get referred to PCIT if their kids are acting out a lot. There is a very large evidence base for PCIT in diverse groups of families. Internet-based PCIT and PCIT in general seems to increase sensitive and responsive parenting and reduce childhood behavior problems. Given that there's this powerful evidence base, it seemed to be a good intervention to see if there's actually potentially biological impacts of PCIT. Internet-based PCIT is the same as PCIT. The big difference is instead of live coaching from a separate room through like one of those cool mirrors, you are live coaching through a computer. So the parent has some form of Bluetooth speaker, headset in their ear through which the therapist is coaching them to increase their positive parenting skills and decrease their negative parenting skills. So we are having parents learn how to better praise, pay more attention to things that are more positive or socially promotive in kids, and then actively ignore behaviors that are problematic. Parents also learn consistency, which is a really key factor for them, especially when they are showing up for therapy. PCIT is a really powerful intervention. I'm trained in it and I've done it a bunch, and I find that parents respond really well to it and end up describing a lot of a better connection with their kids when they're done. I've heard from multiple parents that while they have always loved their kid, at the end of PCIT, they end up saying things like, oh, I really like my kid again now, or how we have such a better connection. So it's been powerful to observe, both reading about it and implementing it. So let me reframe to the specific particulars of this PCIT study. For the epigenetic age study, we were recruiting from a larger ongoing PCIT study that was originally designed to test the efficacy of internet-based PCIT in a sample of kids with developmental delay. In South Florida, where the kids were located, there's a certain series of state policies, and at 36 months, kids with developmental delay would actually age out and be in a gap for receiving these services. So the researchers identified this gap, and we're trying to see if PCIT might be a way to fill that service gap. This is a really important group to study, not just in general, but for this question, because they are understudied. Kids with developmental delay, we're seeing increases in prevalences of developmental delay. It's pretty common. We see more developmental delay in the context of more stress exposure. And there's a good evidence base for PCIT in the context of developmental delay. Another really important point about this specific sample is that from the location that they were being drawn, they were half English and half Spanish speaking. They also, in order to be enrolled in the study, they did have to exhibit clinically significant behavioral problems. So we ended up with a sample of 62 children who were pretty much exactly 36 months old, so 3 years old, because they were aging out of this certain service. 70% of the kids were Hispanic white, and 71% of them were boys. Very consistent with kids with behavior problems. You usually see higher rates of boys. Most of the parents who showed up with their kids were moms, also consistent with the research. The sample experienced a fair amount of stress. 65% of them reported significant impairing economic stress. So this is a study where we're trying to look at adversity in parenting. So we did want to quantify stress exposure. We used a cumulative risk scale. So cumulative risk scales quantify the presence of multiple co-occurring stressors, because there's literature that shows more stress exposure by count is associated with more problems. It's a concept that's very similar to ACEs, or adverse childhood experiences, but because ACEs are a specific type of stress to which children are exposed, cumulative risk, the actual stressors that you measure tend to be a little bit different. So some examples of stress that were measured in this sample was single parent status. We also looked at low income, and another example would be parents reporting clinically significant levels of depression. The range of our cumulative risk scale was 0 to 9, and on average, kids were reporting 2.5-ish cumulative risks, and here's a histogram showing the actual distribution for the sample. So kids are reporting notable levels of stress. We also measured positive parenting, and we measured it really rigorously. We used a traditional measure in PCIT, which is the dyadic parent-child interaction coding system. And the key part about this is that it's observed parenting. So a standardized observed measure of positive parenting where trained coders came in and counted and quantified the display and proportion of positive parenting behaviors. I really am a big fan of observations in my research, because it really helps us drill down to really objectively see what is going on in the dyad. Okay, let's take a step back, put this in the timeline because it's a little bit complicated. So first we have randomization for that big study, ended up being like 150 people. People were approached regarding our secondary DNA study. We got a small subset of folks who were interested between, we had 62 folks, 30 of which were actually in the intervention condition. Then we had our baseline visit where we assessed parenting using that wonderful observations, and we assessed adversity exposure. Then there was the intervention or control period. Control was referrals as usual. This took about 20 weeks. On average, kids completed 9.5 sessions, range of 0 to 19 over those 20 weeks. Then immediately after the intervention, we had our post-treatment. Again, we assessed parenting. This is really important. So we have 2 measures of parenting. So when we are looking at changes in parenting, we have our baseline and our post. And then a year later, so kids are now, they go from being 36 months or 3 years to about 4.5, because we have the intervention plus the year. We took saliva to get our DNA methylation. Saliva was used to assay it to get DNA methylation levels, which then was used to calculate the epigenetic clock. And there's a bunch of different ways to calculate the epigenetic clock. What's really important here is we use the pediatric buccal epigenetic clock, which is a relatively new introduction to the literature and much better in pediatric samples because it was actually trained on pediatric samples. So of course, the first question is, were there main effects? And unfortunately, at this point in time, we did not find any. So what I have behind us here is a box and whiskers plot to just show you what the data look like. We have on the left here our PCIT group, on the right our control group, and what you see here is 2 different plots that are not statistically different. I show it to you because you can see that things are kind of going in the hopeful hypothesized direction. Children in the PCIT group are displaying 0. If you look at that y-axis, they are looking like they have an epigenetic age acceleration of 0. Higher is what we would think of as potentially problematic. So maybe there's something going on here, but the stats suggest we are either underpowered or there isn't a statistically significant effect. And that's not surprising. We had a small sample. So we also wanted to look at the data another way. Another of our hypotheses, as I had talked about, is that parenting actually buffers the adversity and epigenetic age acceleration association. So what we did here is to optimize the variance in this context is that we actually collapsed across groups, and we, instead of looking at intervention, we looked at change in parenting. So from pre to post, how much positive parenting were the families displaying? One of the reasons we did it this way is, on average, if you look at the primary intervention trial, which is published in JAMA Peds, if you are curious, positive parenting did increase in a significant way in the intervention group. So again, we couldn't really test the effect of intervention with this, but at least we can maybe look at the mechanism of the intervention, that caregiving dyadic mechanism. Okay, let's take a moment here. Interactions are always a little tough to go through. On our x-axis, we've got cumulative risk. Higher levels of risk means kids are experiencing more stress. On the y-axis, we have epigenetic age acceleration 1.5 years after that baseline. Higher levels of epigenetic age acceleration is usually what we would think associates with poor health outcomes. And then we're going to let's look at, the interaction was statistically significant. I've got that data up there if you're curious. Here is the low positive parenting group. So again, this is low positive parenting at post, adjusting for pre. So this is the group that showed the lowest levels of those positive parenting skills, like praising, reflecting what their kids are doing. So that line is pretty flat, both statistically and visually. What it looks like there isn't really a link between risk and epigenetic age acceleration. Now we have the average group. In actuality, this average group showed slight increases in positive parenting. I say this because the sample was an intervention sample. So on average, we actually saw if you collapse across the kids, a little bit of an increase. So this is adjusting again, remember, for pretest. Again, though, there's no statistically significant relationship between risk and epigenetic age acceleration. Now, what's really exciting is this high positive parenting group. So in the context of high positive parenting, so at Post, these are the families that are showing the highest levels of praising and reflecting and noticing the positive behaviors their kids are doing compared to their pretest levels. In the context of high positive parenting, high levels of risk were actually associated with the lowest levels of epigenetic age acceleration. So one thing I want to show you is if you look at this graph at low levels of cumulative risk, there really isn't any difference in epigenetic age acceleration. All the action is happening at the high levels of cumulative risk, and that the statistically significant relationship is only happening for the kids who are exposed to high, like more increases in positive parenting. So when parents of very stressed out kids markedly improve their parenting, these kids showed the lowest epigenetic age acceleration, making us think that this might actually forecast potential for improvements in health. Maybe. So to sum it all up, what we see here is that increases in sensitive and responsive parenting might actually attenuate or disrupt the effects of adversity on a measurement of biological age acceleration. And this study is published in Psychological Science. Here's a QR code if you're curious about it. I think it's pretty exciting, and I'd love it if you wanted to look at it more. Okay, so that's PCIT, a great intervention, but we didn't find a main effect, kind of underpowered. But we found some really promising results suggesting that maybe parenting is important here, changing parenting at least, for kids' biological aging. I then came to UCSF to learn more about developmental psychobiology and how these psychosocial interventions actually change kids' bodies. And one of my first incredibly fabulous opportunities, I was so excited to be able to work on this study, was to collaborate with an all-star team to look at maybe if child-parent psychotherapy had something to do with kids' biological aging. And there are a lot of you here right now, as I put up the slide on child-parent psychotherapy. So I'm going to be brief in how I explain it. A lot of you are very familiar with this therapy, have seen how powerful it can be for kids. The majority of the research is conducted here. So child-parent psychotherapy is a manualized therapy for young kids and their caregivers who have been exposed to trauma. It was co-developed by Dr. Alicia Lieberman, who is here at UCSF, and Patricia Van Horn. The intention of this intervention is to improve psychological and relational functioning of the dyad, of the child-caregiver connection. It's rooted in attachment therapy, and its central target is that relationship. Child-parent psychotherapy targets inside of that dyad an enhanced sense of safety, particularly for the kid, improving parenting self-efficacy, improving the caregiver's capacity to reflect and empathize with their kid, and improving and promoting adaptive coping for everybody. The sessions take about a year, and the sessions use play-based trauma narratives, unstructured developmental guidance, and reflective prompting to teach these different treatment targets. The evidence base for CPP is multicultural, it is bilingual, and it is international. And there is a ton of research for CPP ongoing right now. And the research continues on the second floor of this fabulous building. There's a ton of evidence for child-parent psychotherapy. We've got 5 randomized controlled trials across 2 different sites, evidencing strong improvements on child health. Child-parent psychotherapy reduces kids' traumatic stress symptoms. It reduces their PTSD, their behavior problems. It promotes or reduces caregiver avoidance. It reduces caregivers' child-rearing stress, and also just improves their affect, caregiver affect, another really potent predictor of child outcomes. And it increases the likelihood of displaying secure attachment within this dyad. So CPP has this fabulous and strong evidence base. Again, this makes us think this is a good intervention to try and see biological change. Bringing us to the question, can CPP slow kids' biological aging? I was part of a massive collaborative effort here to test this question. And I'm going to explain to you the intervention groups and how those data were collected. We had our intervention group, kids who experienced or who received child-parent psychotherapy. 45 of them, they were young, and 76% of them were Latino. This is the lovely team who collected these data. I think some of you are here or on Zoom, including Dr. Alicia Lieberman, and we've got Luisa Rivera, who is essential in supporting the data collection and increasing the comfort of the families in engaging in the study. Something I want to highlight about this study is it was quasi-experimental. We did this so that we could have causal inferences without having to randomize kids to a treatment that we know improves their trauma symptoms. So in this quasi-experimental study, we had our comparison group. So kids weren't randomized, but rather we compared them to a matched comparison group of the seed cohort. Another sample that y'all have probably seen a few times, led by Dr. Nikki Bush and this team of 110 kids, similar age groups, 36% Latino. Key point here is data were collected before treatment or before a waiting period, and then 10 months later. So we have repeated measures of epigenetic age acceleration. These data are there. There's no other study out right now that has these data, pre and post. And we took buccal swabs, which are cheek swabs. So we had these huge teams collaborating, and then we brought in another team, which was Michael Colbor's team at the University of British Columbia, Vancouver, and Dr. Sarah Merrill, who is the co-first author on this publication, which is currently under review, who led our data analytic and curation efforts, which is a huge task. We're dealing with trying to turn specimen into an analyzable number. So we collected buccal swabs at pre and post, and we used those to calculate epigenetic age acceleration, again, using the pediatric buccal epigenetic clock. As I mentioned, this is a quasi-experimental design, but this is a sophisticated quasi-experimental design. We matched. So we used weighted propensity scores with matching, with weights to achieve balance across the cohorts so we could compare them confidently. And the seed cohort and the CPP cohort, as I described to you previously, have a lot of similarities. One thing I didn't express, which is really important, is that both of them are exposed to high levels of stress. They are good cohorts to be able to compare. And we made that even stronger by matching. So let's look at some pre-data. We've got on the left our child-parent psychotherapy group. On the right, we have our community sample. Similar construct for the other graph with epigenetic age acceleration. On the y-axis, more epigenetic age acceleration means faster accelerated aging, what we would think is problematic. And the critical point is that these groups were well matched. Epigenetic age acceleration is not different, and it rests at about 0 for both groups at baseline. As you can see, that difference is not significant. So here is where the cool finding is. At post. So after receiving treatment or going through that waiting period, we do see a difference. The child-parent psychotherapy group, their epigenetic age acceleration is roughly the same. If you look at the graph, it rests still at around 0. In the community sample, however, their aging continued to speed up. As I described to you earlier, that's what we would expect. Stress-exposed children, they experience accelerated aging when there's nothing that happens to buffer them from that. What we're seeing here is experiencing or intervening with these kids with child-parent psychotherapy seem to slow that process and potentially protects these kids from the biological consequences that we know are associated. with faster biological aging. So what we found here in this incredibly novel and exciting study is some of the first evidence, especially using pre and post data, that exposure or participating in child-parent psychotherapy, a dyadic intervention known to support the parent-child relationship, seems to protect or buffer kids from accelerated biological aging. Okay, we have a couple really cool findings, all coming together to potentially suggest that there's something about this dyad improving parenting and giving kids this effective therapy that might be protecting them from accelerated biological aging. But what does that even mean for a sample that is like a child sample? Children are supposed to develop. They're supposed to age. That's what we would expect. So one thing that's important to note is there is that evidence, and the evidence is growing and it is becoming stronger, that adversity exposure speeds up aging in kids. Speeding up aging is actually likely adaptive. So if a kid is exposed to a lot of stress, it would make sense for them to access more of their physiological resources, run their system a little harder in order to cope with all that. It would help them be able to deal with the things that are being thrown at them. However, as we've talked about, nothing really comes without cost. Accelerating someone's aging likely comes at it's taxing to your system. And that may be part of why we see these health problems for these kids down the road. They have to work extra hard. So we think that these findings are really promising, right? We think that they are suggesting these kids are going to be healthier. And we think that this is an indicator of a biological signal of a psychological intervention. But we really need more longitudinal science, both in interventions and in prospective cohorts, to better understand these mechanisms of action. So to summarize, early life adversity gets under the skin, and it seems to show up as health problems for kids who are exposed to a lot of stress. What we have here is evidence from 2 samples of children who have been exposed to high levels of stress that seem to evidence that parenting or protecting or supporting parent-child relationships seems to protect kids from the biological embedding of stress as measured by accelerated aging or the epigenetic clock. We have the IPCIT findings and internet-based therapy that shows that increases in positive parenting attenuated that risk and aging association. And then we have this really exciting under review main effect finding that just participating in this amazing intervention seems to slow down that aging process in kids exposed to stress. So what we think here is that psychosocial interventions that target parent-child interactions, we know that these are incredibly helpful for kids' mental health, for parents' mental health, for the dyads' relational functioning. This body of evidence also seems to show that these psychological interventions, psychosocial interventions, change kids' bodies in ways that we think show that they're going to be healthier. So let's work to make it easier for families to access these interventions. All right, thank you so much, everybody. I wanted to just put up a sliver of the acknowledgement slide that of all the individuals who contributed to all of the science, and a really special shout out to Dr. Nikki Bush, who has been a huge guide to me in putting this story together. And here is a place where you can find more information on the studies and our fabulous website of all the research that we're doing, and a recent talk that I had about the IPCIT findings. Thank you.
That was amazing. That was fantastic. Thank you. I want to make sure that we get questions from everyone. We have a sizable group in person, and I'm going to reverse it. I'm going to take people who are on Zoom first, and then people in person. We'll be reading out the questions for you, Dr. Sullivan.
Awesome. And are you reading them, or should I repeat them? Okay.
All right. Thank you so much. The first comment. Could you please review what we know about the longitudinal course without intervention and in the general population of such epigenetic changes first detected in childhood?
Okay, and should I repeat that to the, because I want to make sure everybody can hear it.
Yeah.
Okay.
Let's do this together.
Yeah, that sounds great. Review the longitudinal course without interventions in the general population of epigenetic changes first detected in childhood. So the current body of longitudinal evidence is, first of all, we would love more. But there's growing longitudinal cohort research that does seem to show, when you look across the science, that increased exposure to adversity seems to facilitate what I'm more of an expert in the clock, less on the actual the specific CPG markers, but it seems to be related to increased epigenetic age acceleration. And there is a few longitudinal cohorts. There's a couple of these papers in JCPP that show that across time, kids who are exposed to more adversity seem to show accelerated epigenetic aging. And then there's only a couple, but they're really cool, that show that this acceleration then is related to prospectively increases in problems. So like internalizing symptoms.
And we'll do one more from Joan, wondering about positive.
Now they can hear, whoa, now they can hear me? Okay, in Zoom land? Okay, Joan, you can go ahead and unmute yourself and ask your question about parenting observation with the CPP study.
Yes, I found it really fascinating. As you disaggregated in the IPCTI study, like it was actually those who changed their behavior and were observably doing more of the positive parenting behavior, that actually seemed to show more of the change. It was that subset of people. I was wondering if a similar sort of observation of parenting behavior was done with the child-parent psychotherapy study. That was my first part. But I guess more to the point is like, what do we know about what actually drives this change in parenting behavior? Were there like parent characteristics that they brought into the intervention, right? Was there something about the intervention delivery or the degree of rapport with the clinician? Or was there any insight there as to like why some parents responded like so much more with like more observable change?
Great question. Okay, so, and I can speak a little more confidently to PCIT because of me practicing that. So drivers of change in parenting intervention, certainly rapport with the clinician. I think we just know across the board that like having a higher rapport with your clinician, regardless of psychological intervention, improves effects. In PCIT, they tend to think that it's the coaching and the increased attention and following of parents. For instance, in PCIT, what we know is you basically will progress through different exercises with a coach talking to you the whole time while you're playing with your kid for like half an hour. And they will continue to prompt you and then track. They'll do kind of a pre and post test of how many more positive parenting behaviors you're showing. And so they associate that use of that coaching as well as parents' actual practice, parents who practice the skills and generalize them more into their home environments. That also accounts for how we see changes. Another thing that I guess I didn't highlight in the IPCIT study is that certainly there are factors that make it easier to engage in parenting. We still saw that biggest effect in the families exposed to the most stress. So families exposed to a ton of stress still had the capacity to really show up for their kids. So what I'm trying to say here is certainly some of these stressors can actually get in the way of being able to engage in therapy. But what we saw in that actual study was the parents' capacity to use these skills in the context of a lot of stress that really protected their kids.
In the audience?
Here we go.
Thank you so much for a great talk and for all the work you're doing in this space. I'm wondering if you or others have done work that looks at the maybe a more proximal measure of the impact of stress psychologically, like manifest anxiety in the children or the caregivers, and the rate of acceleration or the other markers that you're doing.
Okay, so you mean like acceleration over, like accumulating over time, and the stressors that like predict that slope? Is that what you're speaking to? Or could
That may be a smarter question, but what I was asking was if you have measures of anxiety of the parents or the children pre-post, and whether there are linkages there.
Not in this study, but yes, measures of anxiety of either the parents or the kids pre-post, and how that relates to accelerated aging. They're not in this study, but JCPP seems to publish a lot of this stuff. And there's a couple different researchers, Schenk and Demmering, who have looked at internalizing symptoms more in kids. I haven't read anything in parents yet, but that's certainly something I'm really curious about. And how they'll look at epigenetic age acceleration longitudinally and how that relates to changes in internalizing symptoms. And that's one of those rare prospective cohort studies that did actually find an effect of more acceleration, showing more problems in like internalizing, so anxiety and depressive symptoms.
Great. We have another question from a participant on Zoom. Wait there. Oh, Melania. Go ahead and unmute yourself.
Hi. I was just curious if you could speak to, or if you know about any other studies that are looking into just like the biological underpinnings to everything you described today, such as like which neurotransmitters are coming as a result of things like CPP or the other dyad therapies, and then how that's like signaling other systems in the body to potentially like have the effects you described, like buffering accelerated aging.
Gosh, okay, so totally. There is a bunch of research on this. And one of my favorite studies, I think it's Aschbacher and Bush, like, and a bunch of other people, a huge group with CPP in, I think, 2021. They did a really cool study where they actually found that maternal immune phenotypes was related to treatment response. So mom's immune functioning, so inflammation, right? That seemed to relate to how kids and parents responded to child-parent psychotherapy, such that more pro-inflammatory phenotypes was related to less ideal response to child-parent psychotherapy. And that's just like one study of many. We just did a review of dyadic intervention effects on multi-system biological effects. It's in psychoneuroendocrinology. It's still in the proof stage, so if you look at it, just be gentle. I just gave them the proofs. We did a review of everything we could find that used a rigorous design of randomized controlled trials of a dyadic intervention with a biomarker outcome, and that goes through everything. And there's accumulating evidence, but it's more like 1 or 2 studies per system. So I think if you were to take a look at that paper, that if you're curious about it, it's in Psychoneuroendocrinology, Sullivan et al. 2023, Bush's last author, that goes through a bunch of cool outcomes.
Okay, thank you.
Other questions? Okay, here we go. Jack.
And Allie, make sure to repeat Jack's question, please.
Am I too close? No, here we go. Thank you again for this study. I'm curious about your second study in the results slide. I'm trying to understand if you could help us contextualize the effect size. It sounds like there's still a lot of research trying to understand what the correlates are of accelerated aging, but I'm curious if you can just help us think about how to interpret this finding in effect size.
Yes, totally. When in the write-up, I think it's a small effect size, like the Cohen's, it's not a D, it's an ANCOVA number that is escaping me, forgive me. But this effect size is small. Something to think about with that is that Traditionally, biological to psychological or psychological to biological effects are typically small. The measurement is messy, and going through that many processes can introduce noise and make the signal a little harder to detect. However, so if you just like look at the numbers here, the difference, right? It's like 0.2 like of a year. We're not really able to like interpret, oh, these kids are, a quarter of a year older than the other kids just yet. At least that's not where we're at in the epigenetic clock world. What we are able to understand is that small effects to biology in like a 5-year-old have a tendency to accumulate over time. There's some research in inflammation that shows that smaller effects of pro-inflammatory phenotypes when kids are young add up as they age. That effect size tends to increase. And so this effect, the signal that we're seeing, while it may seem potentially on paper small, we hypothesize that it's going to set kids up for a much more accumulative, healthy trajectory when we think about how many processes these kids are going to be engaging in over time.
I have a question about this slide as well.
Yeah.
Is that okay? Just a clarification, community sample means they were getting an intervention or they're a wait list, or like they're just not.
So great question. It's a seed cohort. So it's a prospectively followed cohort. They didn't get any intervention during this time. They are exposed to stress. And we used essentially fancy stats to match them. So weighted propensity scores so that we could achieve balance. And the weighted propensity scores use things like the trauma exposure score. They use things like child sex, buccal cells, which is a thing that you need to control for when you do clockwork. And I think also we use those factors to make sure that we weighted the epigenetic clock across groups so that we can make a strong inference from this quasi-experimental sample. So good to compare with from the get-go, and then we took it a step further to use weighted propensity score matching.
Thank you. Okay. All right, Sharon on Zoom, you have a question. You can go ahead and unmute yourself.
Hi, what a wonderful piece of research. This is so necessary to help people who still don't believe in attachment theory, or that therapists can speak on behalf of and interpret even a pre-verbal child's trauma. So I really want to send, my appreciation to Alicia and Patty, because without them, I think so much of us would not know how to do this work. And what I want to know is, so when you were doing the work and working with the therapist, and were these all therapists, or did you use how, the program has also changed people who are not people with degrees, but did you get resistance from having to speak about, like what Alicia says, naming the trauma, and understanding for the parent that it matters that the child knows what happened, and also that, sorry, let me just ask that question.
That is an excellent question. And I was not part of the actual in-the-room therapy. So difficult for me to speak to that. And where I think I'd have to point you is to some of Dr. Lieberman's research on how CPP, potential challenges that families have, because I would expect that the same things that come up for clinicians in CPP and other contexts came up in this study. The only difference is that we also took biospecimen. What I can answer, you asked a question about whether it was community providers. The data were collected here. So it was our CPP clinicians here, and I believe in Oakland as well. So the typical CPP clinicians who we know and love today.
That's great. Thanks. And so did you use that? Did the therapist then ask the parents, or was it already collected about the trauma history? And did you get the trauma history of the parents as well as in the situation that they were being treated for?
Gotcha. So did we collect data? Did we collect data on family's trauma exposure? Yes. So we also we matched we use that in our data analysis, a more crude count, at least for kids' trauma exposure. And I think psychologically for the parent exposure, I don't know off the top of my head because we were focused on the kid outcomes. So we were controlling for kids' trauma exposure. But that is something that I could definitely look into and get back to you.
And one thing, I did my dissertation on therapist use of the ACEs, and it's very interesting how much pushback there is about not just the ACEs, but that therapists don't really want to ask about traumas. They want to wait for the story to unfold. And I think that's the brilliance of what Alicia teaches, is let's get to that splinter quick. All right, thanks.
No, that's a great point, and something that I've encountered with practicing a different form of trauma-informed cognitive behavioral therapy, like TFCBT. It can be really difficult to learn how to ask directly about stress to which families have been exposed.
Thanks for that thoughtful response, Dr. Sullivan. Questions in the audience that I can run to you. I'm coming to you. Go.
Hi, Allie. Thank you. Well, less of a question and more of just a note of gratitude on behalf of Child Trauma Research Program, because for many years we've been looking at child-parent psychotherapy, and there's so much evidence with the psychosocial and the metrics, and I mean, it's mind-blowing. But this truly legitimizes relationships as a central force in change, right? And so on behalf of our team, thank you. And just the elegant display and really hard concepts and really in ways that we can internalize. So thank you. And I'll just add a question, just a global question. As we're looking at, we're a clinic, we're here, we still do research. We don't do as much of this biological piece. It really is very rigorous and requires a whole other constellation of factors that we're so grateful that was handled so elegantly by Bush and team. So are there low cost or any other ways that we can continue to integrate elements of this research design to continue informing the field and continue to legitimize interventions that are relationship-based, whether it's child-parent psychotherapy or others? Any recommendations as you have a better landscape of the research that's happening, what reviewers want to see in order to give the blessing that, these things matter and work? What would you recommend to clinics that they might be able to do if they wanted to do some research and continue so that it could continue furthering this amazing field?
Yes. Let me summarize that question. What are our recommendations? I think my voice carries a little more than the mic is used to. What are our recommendations for clinics to keep doing the science that takes biological specimen and then assays them to see how we can quantify the effects of relationships on kids' bodies? So there's a lot of movement in the tech on collecting data. I mean, I'm looking, I have a wearable, like how many of y'all have a wearable on your body right now? This one's really good. It gets heart rate variability. And the issue that happens with specimen is that collecting the specimen is one thing. Assaying the specimen can be pricey. So I think a step is to see if there are ways to turn the actual biological specimen into a number we can put into a database. We need to work on making that more affordable because there's these amazing blood collectors that I think they're called like the Tassie. I have to look it up, but like it's easy and it doesn't hurt and it takes just a tiny little bit of blood. There's mechanisms like dried blood spots now, which blood is really like the go-to biospecimen that people like these days for getting biological data from. But again, the issue is as we're getting more shelf stable and easier access to specimen, we need to figure out ways in which to more effectively, cheaply, and reliably turn that specimen into a number to plug into a spreadsheet, because that's the coolest and also weirdest part as a scientist, where we're like, I've watched them take the swab and then it ends up as a number on my like SPSS or like R file. And I'm like, oh, that is, how many steps did that have to go through to become a usable, testable number? So I think my primary recommendation is, let's see if we can make that body part to number pipeline a little more accessible, reliable, and cheaper.
That's amazing. Thank you. Wonderful, wonderful talk. You have to come back and tell us more about your next projects. That concludes today's GR. This was lovely to see everyone in person and fantastic showing on Zoom. We'll tell you later how many people showed up. It was a lot. All right. Thanks, everyone.
Thank you. Thank you so much.